Hi, Robert. Good to see you again.
Oh, hey, she.
Hello.
Hey, how's it going?
I'm good. Things getting better here in China, but I saw the news.
No, yeah, that's good to hear. Things are improving in China, and things are just about to get started here in the US.
Okay, I'm really sorry to hear that.
Now, I mean, I don't want to go into politics, but we'll get over it. It just takes longer than it should have.
Okay, just stay at home.
Yep, that's what everybody's doing right now, at least in most of the states. And this cough was not disease related. I got something in my throat.
Hmm.
Let's hope so.
um, yeah, no, great that you could join. Great that you can make it. Did you attend the webinar on Tuesday by any chance?
Yeah, yeah.
Nice.
The panel design
Yep.
is great. It's great. Because we got more details about how panel design.
Mm-hmm.
I think it's useful. Because sometimes the details, our picky researchers, they quite picky about this kind of thing, the parameters and metrics. Some even the algorithm, they always struggling with like the false positive
Right.
rate, and how did you get this false positive rate? I said, I don't know. I might organize something.
Yeah. No, that's
Hello, Robert. Can you hear me?
yes, we can.
Yeah, I'm Ren. Nice to see you again.
Hey, Ren. Forgive me. Where are you from again?
You didn't remember me?
Oh, Prisma?
Huh? I didn't get. Yeah, I you Ren. You didn't remember? I write you emails all the time.
Oh, sorry, Ren. So you just emailed me 2 days ago?
Yes.
3 days ago?
And
Sorry
you
about.
reply. Yeah.
Yeah, no, I do remember you now. I just get a million emails every day. Great that you
Millions
can
of
make it.
emails every day. Sorry.
I think I responded to your emails are very, very infrequent, but very, very long and very detailed.
Yes,
And
because now we are reaching out to the customers recently, and I have to give several talks in the next few weeks too for the mission bio. Yeah, I think for the new year 2020, we might have several customers very interested in this part, but now they are quite interested in the new function of mission bio, the CNV detection and the protein detection especially. So I read you the email last time that we probably need to arrange for a webinar or a call for these things in the detail. And for the materials like the slides to show the product and the technical details of these products and how to analyze this data would be even nicer if we organize for a training of this part.
Yes, absolutely. We are definitely committed to supporting you. You know, questions you have around the workflow or the analysis, I think it's a good idea to have a meeting to talk about this more effectively. We are still building the documentation so that we are able to give you all the collateral so that you know how to use the product and how to best extract meaningful information from it.
All right. So maybe for now, we first arrange for a technical report or technical webinar, something about the CNV and protein expression principles. Do you think it's okay?
Yeah, yes, I think so. Let me just check with Hannah really quick. So I think Hannah, do you want everybody to introduce themselves, or should we just go ahead with questions as they come in?
We can go ahead and welcome everybody to the first session for the APAC region for office hours, and Robert is hosting today. And yeah, we can go ahead and just go into the questions as people have them. It looks like we've got a good number of people that have joined, and I think many of you did join us on Tuesday and had some questions there for Jim that he wasn't able to answer. But you know, Robert's also the expert, I think, with a lot of the questions that you had. So please feel free to go ahead and either write it into the chat, or we can go ahead and unmute yourselves and we can just ask Robert directly if you've got questions.
Great. So that's a yes. Ren, you can go ahead.
Yeah.
Okay. So now we are writing some documentation of mission bio to show it to the customers and to the agents. So now, when customer buy the instrument, is the Tapestri DNA gasket and base plate and something else is included in the instrument.
Yes. So if I got the question correctly, each instrument comes with its own base plate and with its own cartridge that is required for self-installation. Does that answer the question?
The cartridge is a kit, right? We have a DNA cartridge kit.
Correct. And so in addition, as soon as you, once you purchase reagents, either our standard 8 reaction reagents or our installation reagents, which are compromised of 2 reactions, they will always come with reagent kits as well as with cartridge kits.
Okay, so make it clear. If the customer first buy the mission bio instrument, they will have the instrument and every accessory partials for it, like the gasket and base plate. They will have also the kit for 2 reactions, right?
Not automatically. So the instrument itself is an item that you can purchase, as well as the installation kit, which most customers purchase alongside.
Okay, so the installation kit is not shown in the protocol we got, so we have no idea of this part. The installation kit will compromise the DNA gasket, base plate, and the reagents for 2 reactions, right?
That's correct. The installation kit is essentially identical to the reagents that you would receive with a regular kit, only that instead of including 8 reactions worth of reagents, it includes 2 reactions worth of reagents. And the panel that is included is the AML panel.
Okay.
Hi, Robert. Questions. The kits, the standard package of 1 kit, we can use it to do 8 samples, but can we apply it at the same cartridge at one time?
That's correct. Right now, at this point, every cartridge is a disposable item that can be used one time for each biological sample. And each kit that you purchase is coming with 8 individual cartridges plus 8 individual gaskets. Does that answer your question?
No, it's not my question.
Okay, sorry.
I know it's disposable and it's for the cross-contamination. My question is, if I have the samples from 8 patients and I want to apply at once at only 1 cartridge, I want to mix with 8 samples and I want to barcode it with the sequence you offered on the sample tag. Can I do it once?
So I think, hopefully I got it this time. So I think what you're describing is multiplexing multiple biological samples into one, and then loading that sample onto 1 cartridge in order to process that on the Tapestri platform. Is that correct?
Yeah.
Okay. So that's, although technically feasible, and there's nothing that speaks against doing this because the platform would be able to support that. it is going to be a challenge for you bioinformatically to demultiplex your individual biological specimens, particularly if you do not have orthogonal data that you can use in order to help you with demultiplexing. So to give you an example, what you describe is essentially, in a very basic sense, a patient that has undergone transplantation and now has a genome from two specific specimens, one from himself or herself, and then one from the recipient. So you could use our panels. It doesn't really matter which panel. You will always collect and detect germline variants that are not necessarily disease relevant, but they're always part of the panel that you purchase. Assuming that every specimen is very unique in regards to germline variants, and every patient or every individual exhibits a very unique signature, you are able to use our pipeline to detect those differences. We do not support that with our current Tapestri platform to automatically detect those individual specimens. I can give you I think you may be aware of the publication, one of the earlier publications out of Stanford that was part of the beta program from Jim Zender's lab. This is, I think, a nice illustration as to how the system is able to detect individual species that are loaded together on the platform. But as far as adding multiple samples together in order to save cartridges, we don't support that. In particular, also, we don't support increasing the cell concentration. So even if you have, let's say, patient 1 and patient 2, you want to save one cartridge, you could conceivably just add all cells together in a 50-50 ratio. Let's say you have orthogonal data available, sequencing data, bulk sequencing data from both patients. You load your minimum requirement of 100,000 cells onto the cartridge, and you just follow the protocol as is. There's nothing different compared to a regular workflow. The difference really comes into play when you try to analyze the data, because now you have about 50% of the cells that you receive will be allocated to one patient, 50% roughly to the other patient. However, you would have to have your own bioinformatics pipeline to separate those into two different data sets and then continue with the secondary analysis. Does that make sense?
Yes. So what I understand from your previous explanation is the tricky part is about like the downstream bioinformatics thing. Am I understand right?
Correct. Yes. Bioinformatically, we have not validated, the robust detection of individual species-specific cells that we call. Again, we don't see at this point a reason why it should not work, but it really depends on a number of different things, starting with your ability to provide sort of the ground truth data to use as a reference to allocate specific cells to a specific specimen.
But
I see.
I want to add something to the question to Liu Shi. The SMK means to the sample multiplexing kit is from the competitor. They can use it to label samples from other sources with the antibody conjugated with the oligotype to tell in the bioinformatic phase where they come from. So for our SMK sample multiplexing, that means in the workflow you're shown here, we can work in 1 week, several samples processed in 1 week, right? Maximally 12.
Yes, and so I want to make sure that we are on the same page. So this illustration outlines a experimental design that most of our customers implement these days in an attempt to maximize sample throughput. And so if you have one operator and one instrument, even though we only support one sample at a time, you can process up to 12 samples in 1 week by following this staggered workflow. We do not have any multiplexing kits available. With the DNA plus protein workflow, we also do not support at this point cell hatching, what is typically referred to, the ability to sort of overload your cartridge and using antibody or sort of phenotype data to demultiplex and identify potential doublets. Right now, we do not support this type of application. So I think that's important to understand. If you only refer to a DNA workflow, and again, if you want to mix multiple samples together, we have had those requests in the past. I don't think any of our users have attempted that. But as long as you are aware of the limitations when it comes to cells per sample and the backend and the ability to process and analyze your data, then you can go ahead. Does that make sense?
Another question.
Yeah, great.
I saw something called the genotype plus phenotype in one single cell. That means the genotype means SNVs, CNVs, and indels detection from the one single cell. And the phenotype means the surface protein expression profiles in the one single cell, right? So that means this phenotype plus genotype in one single cell.
That is correct.
Okay. Another question is about the parameters we want to show to the customer. Sometimes they want to compare it with the competitors. So like how many droplets would be generated during one reaction of the cartridge, and how many beads on the cell barcode to tell the differences of the cell. You know, the oligotype conjuncted to the beads, how many types?
Right.
Yeah, that's the number of metrics.
Yeah, so some of the data is proprietary. So or I don't even know because they're really protected. In terms of the number, so I think it was a multi question. The first question, how many drops do we generate? So as you remember, our workflow includes a two-step process.
Yeah. Yes.
And so the first is very much in line with other commercially available platforms, and that just follows standard Poisson distribution. So it's an encapsulation step that takes about 5 minutes, and you generate about 1 million drops, of which roughly 5% or so are now occupied with a single cell. In terms of the beads, we do which then comes into play when you follow with a second step, which we call barcoding. Beads are now combined together with your single cell lysates. We ensure the instrument itself, as well as the cartridge design, ensures that there is very, very minimal chance of multiple beads ending up in one drop. So that's a very defined metric internally that we call bead loading, and that's kept at a very conservative number to minimize the ability of having multiple beads in one drop. And then in terms of how many oligos or single-stranded DNA are conjugated to the beads, I don't have a number, but it's saturated to the effect that it does not inhibit the amplification reaction if that drop contains an individual cell.
You refer to the multiple beads here. So is there any metrics showing that like the output of the cells correspond to the multiple reads? Like 1,000 cell will get a certain percent of multiple cells, and maybe 1,000 cells, which range would be the multiple reads B. Do we have such metrics?
So let me just double check. So your question is are you referring to titration experiments where you load a certain number of or concentration of cells at different concentrations and you measure the occurrence of doublets?
Yes, exactly.
Okay, so we have limited, so we have done this in the past extensively. I'm just trying to pull up one graph here. So this you're probably aware of, you have probably seen, oops. Just open this up. So I'm at this point, I'm unaware of us having performed titration series where you input a certain number of cells and then a number of doublets. And this is one of the experiments, at least, where you conceptually can understand how we measure doublets by using genotype information. And so that's different to RNA sequencing experiments where you would just essentially do a two species experiment and calculate the fraction of rates that align against each reference genome. The concept is the same, only here that we use genotype information, where you look at, in this case, I don't know, 8 or 9 different loci. Those are 2 different cell lines. They have a very well-defined genotype profile, and there are a certain number of targets. In this case, for instance, those 3 here that we know have to be homozygous mutant in K562 cells or wild type or reference in the Raji cells. As soon as you now receive a cell or detect a cell that has a heterozygous call made in that specific target, you know that's probably the result, very likely the result of more than one cell in one drop. And so this is just looking at the data at the genotype level. If you look at the data at the variant allele frequency level, you will also see a nice signature that this is not just detectable based on these 3 variants, but actually across all the variants. Our metric is 8% in terms of, sorry, go ahead.
8% in terms of how many cells? How many cells?
Up to 10,000. So because we do, we highly recommend not to load more than 100,000 or more than 4,000 cells per microliter. That's our upper limit. If you can guarantee that your doublet rate or cell mixing rate, how we refer to it, is typically well below 8%.
So this number can be read into the parameters, right? When we show it to the customer.
Absolutely.
Or show it to, okay. Yeah,
so I think this is, if you don't already have that slide, this is essentially one of the slides where here this determined as 8% cell mixing by reaching up to 10,000 cells.
Yeah, this performance, do we have these slides? Can you share it to us? So maybe we don't get the new parameters from Mission Bio since last year from May. So maybe there are updated data of the performance parameters of Mission Bio and we didn't receive yet. It would be better if you could share us with the new version of this kind of slides for us.
Yeah, absolutely. No, we can do that. Yeah, we can definitely share the new slides with you, or the updated slides rather, that include both some of the updated performance metrics as well as newest content around publications or applications.
Thank you.
And actually, so one thing, sorry, give me one thing. So I think one, because I know publications are highly valuable, particularly in the APAC region, because they serve as quite useful sales tools. We have had, since we talked, obviously a lot of customers and researchers working with our products and now starting to publish the results. So I think what will be helpful for you is to also be able to use those moving forward. Yeah. And those were just 4 publications published just the past 30 days. 2 of those in bioRxiv from KOL accounts, and then 2 in quite recognized papers around critical applications such as MRD or treatment resistance.
That's really nice if you show it to us, because next week, Monday, I need to go to one customer, really important maybe.
Yeah, I will share that with you. You have time to go through those
to modify,
data, and feel free to email me if there's anything you need clarification on.
Yeah, okay. For the parameter, also we want to write into the Mission Bio parameter is the cell capture rates we can say is around 5% to 10%.
That is correct.
Okay.
And I think that's also important. if I may add something, you may already be aware of that, but capture rates are always defined by the analyte that you try to detect. And our target analyte is primarily DNA, and therefore we have to split our workflow into 2 steps. You will probably hear from customers specifically in your region that are used to 10x genomics, or really just generally transcriptomics at the single cell level, that their capture rates are way higher, which again is the result of only targeting RNA, which does not require your workflow to be split into 2 individual steps. If you look at other commercially available single cell DNA workflows, their capture rate is comparable, if not even lower compared to ours.
I will talk to the customer about this part too. The other one is I saw the sole source documentation from Mission Bell, and they see the panel when the panel designs, because the sole source documentation is to use to tell the difference about Mission Bell and other competitors, right? Mm-hmm. So there's one sentence means the panels must have panel coverage uniformity of more than 8% and reads on targets of more than 18%, right? 80%.
Yes.
So, but I also see some other introduction about the Mission Bell that the panel uniformity is above 90. So this parameter, do we need to set it to 80 or 90?
So the sole source document is not so much about distinguishing us from competitors, but more about characterizing our technology and some of the unique features. In terms of the panel uniformity, so there's varying numbers. You said 90% or 80%. All of our catalog panels that we distribute, AML, CLL, myeloid, as well as THP, come with specs of 90% plus. If you work with a customer that is interested in an in a custom panel, we have the minimum standard of releasing this only if it's 80% or higher in terms of panel uniformity.
Yeah. Also for the parameter to write on the book, the design for the amplicons maximum, we can write it to 1,000, right?
That's correct.
Okay.
So 1,000. Well, sorry, I was going to say 1,000 using our production Tapestri designer that supports the generation of up to 1,000 amplicons. If you or any of your customers are interested in potential, whole exome-like applications or applications that require more amplicons, we do have a channel for that, which would then be the white glove design that can start addressing those inquiries.
Okay. Also, you talked to me last time, like one amplicon typically harbors multiple targets. Here, targets means different CNVs, SNVs, or indels, like the mutation, right?
That's correct.
Here. Okay.
Yeah, technically every amplicon, let's say, has an average size of 200, or any insert size has an average size of 200 base pairs. You would have potentially up to 200 targets per amplicon.
Yes. So we also talked about the CNV and the protein capacity of Mission Bell, right? Could you later also forward to me some technical support materials about this part, so I can show it also to the customers? Because I don't think I get the newest information about this part too.
Yeah, absolutely. We can provide you with the development protocols as well as some of the early access data that demonstrates the utility of that application in the clinical space.
Also, there's something like the after sale support. So if our customer has bought for the machine, the installation should be done by us or by you guys. Basically, we have only done this experiment for only one time, as you know, last year when you came here.
Right. No, I mean, that's a discussion probably we should have offline, but we obviously are committed to making you successful. So once you are in a position to have a customer trained on a new or existing instrument, we are more than willing to be on site to help you and get you up to speed. But ultimately, long term, you should be able to support your customers in your specific territory.
Yeah. I see the new V2 protocol of mission bell is only V2 regions is sitting now, or right? So now everything sold out as V2 regions.
That's correct.
Okay. So we should use dual index PCR for the V2 protocol. So now the library should be different than before, right? Before
That's correct. Yes, the library is slightly different and has 2 indices on both sides, as well as a standard P5 site. So you don't need a custom sequencing primer anymore. Tapestri libraries can be considered as regular libraries that do not require special treatment when setting up the sequencing reaction.
I think this has, oh yeah, one of the questions from my colleague, she asked, could you explain to us how this can solve the problem that, like the CAR T cells development kind of application? Because we are not quite familiar with this field. It would be nicer if you can explain to us how can this mission bell solve the problem, like the CAR T companies or development companies are facing now?
So that's a good question. That's something we've received in the past repeatedly. There is some early work on the transcriptomics side and how CAR T in the personalized medicine setting can benefit from single cell research. We at this point are not in a position to actively support these type of applications. Not mostly, but partially because it is linked to the ability to look at specific T cell receptors and T cell receptor rearrangement. Those are challenging tasks bioinformatically that right now we're not equipped to do. So it's on our roadmap, and we are aware of its impact in the market space, but at this point the products do not directly address the needs.
Okay. So for now, the mission bell is specifically to the cancer research. For the CAR T cells and the other immune cells, maybe now is not feasible for now, right?
I mean, I'll always phrase it, it always depends on the question that you ask. So I would not categorically exclude any CAR T cell application. Beyond cancer, we are also making a move in the gene editing space where it is critical that you, as soon as you are thinking about putting this technology into the clinic and develop an assay that is used clinically, false positive and off-target and on-target rates are critical and paramount. And so to date, there is no platform out there that does this reliably and with high enough throughput. So that's something we are certainly well equipped and positioned to start addressing. And we're here in the US, we're working closely with the NIST government arm, the National Institute for Standards and Technology, to be on the table to decide what is needed and sort of start setting the standards and the SOPs to bring this closer to the clinic.
Okay, now I see. Okay. So if in the future we have this kind of amplification from our customers, maybe in designing this kind of SOP need a bit from your help. It would be nice if you are around to help us to set up this kind of application for the customers.
Yes, I mean, yeah, I can send you some collateral that just demonstrates as a proof of concept that our technology is uniquely positioned to address these questions. We have a handful of customers working actively on that, and so there should be coming out more data soon to demonstrate its clinical utility. but it's, you know, it's going to be a long way to actually have a product on the market that can be used in a clinical setting. But we are well aware that this is the long-term goal, both from a cancer perspective as well as from the gene editing community perspective.
Okay. My last question. So I have used the Tapestri Insights you might send us last year from Liu Shi, but I think the version is not compatible with the datasets for now. Have no idea why. When I loaded the Loom file into the Insights, it says it's not compatible. So for now, if we have the updated Insights software, could you please send it to us so we can do some exercise and show the data to our customers when we were at the customer side?
Yeah, absolutely. So I would encourage you to visit our portal.missionbio.com, or I don't know if there's a Chinese version.
I've been this part, but I didn't find the place to download it. Maybe because it's hiding somewhere, or because I'm not registered.
If you can access that, all you need to do is go to the datasets section. Yes. Sorry. So okay, two parts. First, you need to download the latest iteration of Tapestri Insights. So it should be version 2.2. That's what
mine is 1.6 something.
Oh, 1.6 is
quite
very
old.
Very old.
Yeah.
So that explains your error that you're receiving. If you
download. Yeah, my website must be something wrong. I didn't see anything like this right panel. Okay, I will try it later. Omit this question. I didn't ask. Okay.
No worries. Download this, see if it works. And it should be compatible with data sets that you downloaded in the past or data sets that you download today from this button.
Okay, I see. Okay.
All right. Great. Thanks.
Yeah. Thanks, Robert.
All right, it's past the office hour officially, but I know there are other people on the call as well, so I want to give them an opportunity to speak up and to ask anything that you might want to ask. Don't be shy. No?
Okay, I have to ask for one of my colleagues. She's shy and she's Yu on the page, as you can see. So she asked, could you please share us about the library information? I think you have shown us the structure of the library, like the P5 read 1, I5 index barcode common sequence. Oh, that's the new library. So could you please share us with the new sequence of the library structure to us? Like what is the common sequence and what is the I5 index? Index is in the protocol. And yes, and read 1, the structure.
Yes, I can definitely do that. So I also at the same time want to reiterate the sort of the importance that I think support.missionbio.com is a very valuable resource where you most likely will find the majority of the answers that you're looking for. So this,
okay.
I just typed in barcode. I wasn't sure if we have the article for that, but I've just typed in barcode in the search field, and then there's an article that says, what is the barcode structure of the Tapestri library? So there's V1 chemistry, and then there's V2 chemistry. And so for the barcode, the beads, you have the photo cleavable element. You have the regular read 1 that's about 36 base pairs in length. You have your first barcode, or the first part of the barcode, 9 base pairs. You have a constant region that varies in size, 14 to 17 base pairs. That's by design because you want to offset the sequences in order to generate more variability on the sequencer to help with the cluster formation. The second part of your barcode is downstream of that, 9 base pairs in size. And then you have your seq-F sequence, which is compatible 15 base pairs to your forward primer. And remember, version 2 chemistry is a 3-primer chemistry. And so if you take a look at how we construct the libraries, this makes a little bit more sense.
Yeah, is it possible for us to get the sequence detail of like the constant region sequence and for the which part?
Yes.
Read 1.
So read 1 is just the Illumina read 1, right? That's nothing special about Mission Bio. The barcode 1 and the barcode 2, that's special to Mission Bio. So I can definitely send you those sequences. You will see them anyway in your sequencing data. Constant region I can also give you. Really, this is only important, or you really need to know that if you want to somehow start demultiplexing the cells by yourself, use an alternative method, and other than our Tapestri pipeline. But otherwise, as soon as you use our pipeline the first time and look at the raw files, you will actually have access to all those all data. But I will send you the sequences for, I have them readily available for BC1 and BC2. I have to get a hold of R&D for the constant region, and then read one again is just a standard Illumina read one sequence.
Okay. Thank you for her. And she has another question.
Yep.
So now the website of Mission Bio is not able to register from here. She was trying it for several times, but still failed for now. So when she click on last button, like created account, then there's no response. So she wants to figure out why.
All right, so just make sure I'm understanding correctly. Your colleague was trying to create an account on the Tapestri portal.
Is it?
No.
Is it?
So if that's the case,
Yes.
yes, okay. So she logs in into the, so she creates an account. Typically, she has to verify the account by clicking a confirmation email. And what you are saying is that she's never received a confirmation email?
No, she can't click on the button created account.
Oh.
Here,
The
below
email
here.
and the password,
Yeah.
and confirm the password, and I have no idea to click the last create account.
Here, the blue button here.
This one?
Yeah.
So, okay, let's take this offline. I think that might be related to some of the network restrictions that you guys are facing with in China. I know when I talked to we went through a similar scenario where this page was not quite working the way it was supposed to work in China. So can you follow up? Oh, sorry, go ahead.
Robert, this is she. I think we have solved it. Because I think we can use the account to log in without the VPN condition. So I'm not very quite sure about my colleagues why they cannot create a new account maybe. So I want to know like how about the other users in China region, they encounter the same thing, or it's just the restriction of like our own.
Right. Yeah. So I want to see how reproducible that issue is. If you can just now go to portal.missionbio.com and you can see those fields, and when you fill them, this blue button turns blue, then it sounds like the issue that your colleague is experiencing is either hardware related to her specific computer, or something else that's unique to her. But if you do this, make sure that you accept the T's and C's. And then you should be able to. Yeah, you should be able, it's working for me. So there doesn't seem to be anything wrong with the website itself. Again, I want to make sure that if you do it, that it works as well, so we can exclude the fact that it's a territory specific network restriction issue. And if you confirm that, then we need to take a look specifically at what's unique about your colleague's computer. Does that sound good? Hello, Ren?
Hello.
She said thank you.
I saw on your website your you clicked the create account, it also no action.
Yeah, so I didn't click it. So if you check, so make sure this box is checked.
Yes, yes, I clicked.
Oh, I see. So you clicked, I see. So you see it. Hold on, I need to use it. So that's, so when you click it, you should see this little circle, and then you should get a page that notifies you that you received a new email. But what you're saying, you don't see that page, and that's exactly what I think she experienced in the past. She clicked the button and nothing happened. But I thought we resolved the issue. So let me revisit this. Let me go through my emails to see exactly what we did. I know we talked. We consulted one of our colleagues from the software side to take a look at your browser settings. So, and then we solved it. So I think this is just a browser setting, a web browser setting on your computer.
Okay, thank you.
Okay, if there are no other questions.
Hi, Robert, I have a question.
Yes.
My question is, what's the advantage of existing panels in Mission Bio? What are the specific advantages of the existing commercial solid tumor panel? Is the detection gene site different? That means how can I show our customer the advantage of a Mission Bio in lung cancer testing?
So you mean the advantage compared to
bulk sequencing?
Oh, bulk
for solid tumor panels.
I see. So the advantage of the THP panel, or really for any panel in that regard, compared to bulk sequencing, is mainly twofold. First, you have the ability to look at this single cell level, and by that you typically get a much higher sensitivity of cell clones as well as variants that you are able to detect. Remember, every sample that you process on our platform results in up to 10,000 cells. And so being comfortable calling up to 10 cells with the same genotype, that represents a sensitivity of 0.1% population frequency. That frequency is well below any accepted standard that researchers and clinicians use in using bulk sequencing or other traditional methods. Typically, their cutoff is 1% or 5%. In addition to the sensitivity, and there are a few papers out now that really directly highlight that aspect. In addition to the sensitivity, you now have the ability to clearly outline the co-occurrence pattern of your individual mutations. So what that means is, as soon as you have more than one mutation present in your sample, which in the vast majority that is the case, you always ask the question, do they co-occur in the same set of cells, or are they separated across different set of cells and make up single mutant clones? Bulk sequencing today is very limited in regards to inferring that correctly. And specifically, if you have very, very low frequency variance, 1% or 2% or 5%, it is almost impossible using bulk sequencing unless you assume a number of different things to directly assess whether or not those individual mutations occur in one clone or in multiple clones. So those are the two main aspects. In addition, you can look at zygosity, that sometimes, particularly in the solid tumor community, makes a huge difference. You have LOH events, so limit loss of heterozygosity, where you typically lose one allele as the disease progresses. That's something we can directly assess and compute and quantify using our technology at the single cell level. And what that really allows you to do is directly establish a phylogenetic relationship that sort of recapitulates the disease progression of one sample, or if you work with multiple samples in the time series. Does that make sense?
Okay. Yeah, okay, thank you. And do you provide the data support, or you provide a statistical comparison?
We do have bioinformatics solutions available, both that enable you to process the raw data, the sequencing data, but then also to be able to extract meaningful information from your data using secondary analysis and tools. So that enable you to, let me just open this one here. So, Tapestri Insights is one of them that is more, that specifically looks at point mutations and insertions and deletions. So if you have a time series or multiple samples loaded, you can sort of follow the progression and the clonal evolution and selection over time. We also have a standalone R packages that enable you to analyze your data more comprehensively for CNVs, for instance, copy number events or ploidy.
Okay, thank you.
And then also I want to point out, sorry, I just want to point out that you mentioned lung cancer.
Yeah.
You mentioned lung cancer, and so although the THP panel is very comprehensive in terms of targets that it includes to address a high number of indications in the solid tumor space, it's not necessarily specific for lung cancer. So if your prospect or your researcher does not find the targets in the THP panel that he's looking for, I encourage you to take a look at some of our pre-designed panels or pre-designed catalog panels that are more specific to specific indications. So for instance, lung squamous cell carcinoma or lung adenocarcinoma. Those are probably more suited for that particular researcher. They are not manufactured yet. However, they have been validated in silico. So if you're interested in using any of those panels, they just have to be manufactured. So it skips the entire design process and can be shipped relatively shortly.
Okay, thank you, Anu.
Are these panels shown here already catalog panels, or they still need some time to be available for all customers? This one, pre-designed catalog panels?
So they exist on paper, they exist in our database. We just need the green light to manufacture them. So typically customers take a look at them, they are fine with the content, and they signal that they are interested in using that. So we just have to manufacture them, and then two weeks later, they will receive them. Some customers also use them as sort of a inspiration and then want to add more content or reduce some content. So then this would be a custom panel process.
Okay. So for this page, the number of targets means the number of the base pairs here. Number of amplicons means the amplicon we design for the targets, of course. And the coverage means like 386 base pairs here, and 85.35 is covered, right? If I understand it correctly.
Not quite. And so this concept of coverage for catalog panels is a little bit, it's a little bit suspect, because in order to create those panels, you have to have a target list. And so that target list was defined by someone. And then if specific base pairs are not part of the amplicons that are generated, it'll be not included in the overall coverage.
Okay.
So in other words, 85% doesn't mean it's a bad panel. It just means whatever target list was used initially, did not include 15% of those initial targets did not end up in the panel. But if you take a look at this panel and it overlaps with 100% of your needs, then you can think of it as 100% coverage for your particular purpose.
Okay, thank you. For the CNV and the loss of heterozygosity detection, the bioinformatic parts, the calculation of this CNV is based on the reads, right? Not based on the breakpoint stuff, right?
Correct. So we use actually both. We really integrate in a very unique fashion as genotype data as well as read data. That's the big advantage or the big distinction compared to other existing technologies out there, because we do indel and SNP calling. We have certain sequencing requirements and coverage requirements, which are extremely high or well. So you typically have 60 reads available per any given base pair, and that allows you now to, number one, accurately define specific read changes at any given target, but it also allows you to very uniquely and accurately define your reference population. And that's the key for any copy number assessment, that you need to be able to assess your diploid population, your population that is not impacted by ploidy, because that's the one that you will use as a reference to normalize against, to compare against, and to divide against in order to reveal your changes in copies of alleles.
So the basic principle of calculation of this copy number variation is based on the reads of the cells that might not mutated as the diploid reference, and then we calculate on the parts we want to, like for example this gene, and the reads of this number of this reads compared to the reference, right? This is a calculation.
Yep, that's correct. I think so. Roughly speaking, that's correct. And if you want to learn more about, definitely, download the package, the R package. It includes a documentation that specifically outlines the tools that we use to normalize and to quantify. And then also, I encourage you to revisit the webinar that we had hosted a while ago, and is recorded, where we describe the methodology a little bit more in depth and at high level. I don't know where it is.
I have seen the webinar, but I think for the beginners of the bioinformatics, because I'm not familiar with R language, so is it possible probably we can arrange in the future for a webinar of showing us how to use this R package in analyzing the demo data?
Yes, most certainly.
Okay, thank you.
Well, Robert, I know we've gone quite a bit over time, and just to the team that's tuned in, we will have another one of these office hours. We plan to have them every Thursday at the same time, and so we welcome you to join then. And if maybe if there are additional questions on the line, we can address them then. Also, Robert's always available at support@missionbio.com.
Great. Thanks everybody for calling in and joining and participating. I expect there are more questions, so as Hannah pointed out, feel free to email or make use of the office hours every Thursday at the same time.
Great. Thank you, everyone. Stay safe.
Thank you.
Thank you. Stay safe.
Bye.
Thank you. Thank you.